The cell cycle is the generation of the cell. It is the time the new cell forms until the time that that cell divides. The cell cycle is divided up into different components: interphase, mitosis, cytokinesis, and differentiation. Our cells cannot undergo the cell cycle indefinitely. They are regulated by a piece of DNA at the ends of our genes, and those bits of DNA are called telomeres. Every time the cell divides, a piece of a telomere drops off, and the number of times the cell can divide is limited. Here's an example of the cell cycle. If you consider that the animal cell cycle is 24 hours long, 23 hours of that cell would be in interphase, and 1 hour would be in mitosis, which is nuclear division, quickly followed by cytokinesis, which is cytoplasmic division. Interphase is a period of great activity. If the cell is a pancreatic cell, it will be producing insulin or glucagon or digestive enzymes. The cells are very, very busy during interphase. In G1, the cell is getting larger, growing more organelles. In S phase, the DNA is being synthesized or replicated, and in G2, the cell is getting ready to divide. Mitosis is the nuclear division, and cytokinesis is cytoplasmic division. The animal has two types of cell division: meiosis and mitosis. Meiosis takes place in the testicles or ovaries, and it results in 4 cells, each of which has only 1/2 of the parent's genetic information. Mitosis is very different. Mitosis takes place all over the body, and it is the way that cells divide. The resulting 2 daughter cells will be exactly the same as the parent cells genetically. Here is a very, very quick video of epithelial cells dividing. Epithelial cells are cells that cover or line cavities. You can see this is within the nucleus of the cell. The nuclear envelope has disintegrated, and these are the spindles. The red are the chromatids, or what you know of as chromosomes. And that's mitosis. Mitosis takes place on a continual basis. However, in order to describe it, we separate it into 4 separate stages. These stages are called PMAT, P-M-A-T, prophase, metaphase, anaphase, and telophase. Prophase, the DNA condenses from chromatin, which is string-like, into thicker, shorter chromosomes. Centrioles migrate to the poles, the spindle apparatus forms, and the nuclear envelope disappears. During metaphase, the spindle fibers attach to the centromeres on the chromosomes, and the chromosomes line up at the equator of the cell. During anaphase, the spindle fibers pull the sister chromatids toward the centrioles. And during telophase, the last stage, the chromosomes unwind to form chromatin, and the nuclear envelope reappears. When you see a picture like this, you need to spend some time investigating it. Begin up here with A, early interphase. The cells are growing and increasing the number of organelles, and so on. So you need to go through late interphase, prophase, metaphase, and anaphase and telophase. Understand what's going on in each of the phases. Cytokinesis is cytoplasmic division. It is taking place toward the end of telophase. The very last stage is cell differentiation, where a cell develops its specialized function. This occurs because certain genes are turned on or off while the cell is actively metabolizing. Apoptosis is a form of cell death that is a normal part of development. It begins in the fetus.